So today we're going to talk about Bedtools, and we'll give you a brief demo on some of the uses of Bedtools to manipulate genomic intervals. And so what do I mean by this? There's a little introduction here on the notebook, but the basic idea is that there's many different kinds of data, genomic data, that can be represented as a set of coordinates on a genome sequence. So this can include things like genes, obviously, but also things like attack-seek peaks or enhancer predictions or conserved regions across the genome, or SNPs, or particular exons, UTRs, parts of genes, basically anything that can be described as an interval along a sequence is what we're going to be talking about today. And so there's many different ways that these interval files are stored. They can be stored as GFF files, which is a common format for, particularly for storing gene models, where preserving the relationship among parts of a gene is important, right? You want to be able to tell that a particular transcript comes from a certain gene, and exons are part of transcripts, etc. A somewhat simpler format that's useful for many things is the BED format. And we're going to focus today on manipulations of BED files in particular. So the BED format is a way of storing intervals in a flat file. I've listed here the sort of fields that define a BED file. So it's just a tab delimited file. You can manipulate it with any code that can work with just text files. It's, at least the sort of standard BED file is not compressed or binary or anything like that. But some of the operations that I'll talk about in a minute can be somewhat tricky to compute efficiently. And so using tools like Bedtools that are specifically designed to do these things fast and correctly can be quite useful. So a BED file is defined, technically it only requires these first three fields. So an interval is just defined by a chromosome or scaffold ID that represents, which sequence in the genome FASTA file this interval is on, and then a start position, which is where it starts, and an end position, which is where it ends. Note that the start position is 0 based and the end position is 1 based. So that means this first base of a chromosome, if you start on the first base of a chromosome, your start position would be 0. And if it's a 1 base pair long interval, so it starts on the first base and ends on the second base, your end position would be 1. And so that means that you can calculate the length just by subtracting the end from the start, but it can be a little bit confusing, especially because GFF files use a different convention. One of the nice things about working with Bedtools is that it just handles all of the math around start and end position definitions for you. Excuse me. You can name your interval in the fourth field. Some kinds of intervals, in particular things produced by peak callers or something like that, often has a score. So that can be listed in the fifth field. Some intervals are strand specific, that can be listed in the sixth field. The next set of parameters are graphical parameters if you're going to display this in a genome browser. And then the last three columns are if you have, say, multi-exon genes where you have a gene that starts in one position and ends in another position, but has 6 exons, and so it has 6 blocks within that gene. Only the first 3 fields here that define an interval are required, but you can't skip a field. So if you want to, for example, include strand, you also have to include a name and a score. You can't just include strand as the fourth column. You can use a dot or some other character to just indicate that you don't want to actually use that field. You just need something to fill the space. So you could have, chromosome start end dot for name, dot for score, and then a strand if you just cared about strand, but you can't just skip around and pick columns. Okay, so let's start by just looking at an example of what a bed file looks like. If you run, oops, why didn't that, for some reason, I was, the thing had no kernel selected. So if that's happened to you, I don't know if I messed it up when I uploaded it or if it was just something weird with my launcher. Right up here, there's a little thing that should say bash. If it doesn't say bash, just click on it and switch the kernel to bash and hit select. And then if you hit play, it should work. Sorry about that. So this is a very simple bed file. It's just a list of enhancers from a study that came out a few years ago that identified enhancers in neural crest cells in humans. And this is just a 3-field bed file. It just has a chromosome, a start, and an end. And I've just filtered all these files to only be chromosome 22 in order to keep them very small. So this is the minimal definition, but of course we could have extra columns out here. And some of the bed files that we'll look at have more columns. Okay, so let's talk about what you can do with Bedtools. So for example, one very common task you might want to do in interval manipulation is, say you want to find the closest interval for each element in one file, you want to find the closest thing in another file. So an example of this might be if you have, say, this list of enhancers, and for each enhancer, you want to identify what the closest gene to it is. There's an option in Bedtools called closest that does just this. It takes a list of intervals in this -A option and a second list of intervals with this -B option. And for each element in A, it iterates through B and finds the thing in B that's closest. So a couple of notes: for everything that I'm showing, I'm going to be piping the output to head using the Unix pipe operator so that we don't get a display of tens or hundreds of lines showing up in the notebook. And also, as far as Bedtools goes, many Bedtools operations use this same kind of A file, B file syntax. And it's always going to be, for each element in A, look through B and do something. So A is the kind of thing that you want to get output for each element in, and then B is what you're comparing it to. And this kind of syntax will show up again and again. Okay, so let's see what happens when we do closest. So a few things you can see. You can see that the output of this isn't actually a bed file. It's just 2 intervals stuck together, right? It's giving you for this element in A, let's find the closest elements in B. And in fact, what you're seeing here is that there's many things listed because there's more than one thing that is, if there's ties, if there's things that are equally close to each one of these elements in A, it will list all of them. And we'll come back to this in a minute. So we can reverse the order of A and B. So let's say now we want to take for each gene in this gene UCSC genes chromosome 22.bed file, let's find the closest enhancer or the set of closest enhancers in this file. And if we do that, you can see again that for each one of these, oh, this seems a little, right. So for each one of these genes, the closest element is the same one, right? Because for this gene here, this coordinates, right? The coordinates start much below, they're much smaller than this coordinate. So this is the first of these enhancers, this one that's 1767957. And so all of these genes, the closest enhancer is this one. So each element in A appears only once in this case, because for each one of these, there's only one closest enhancer, but the elements in B can appear many times. They get repeated. So let's say we want to list not just these closest options, but we want to list how far away each one is. Right, so now we're listing for each one of these the closest element in, for this element, we're listing the closest thing and also the distance. And now you can see why all of these are repeated, if it wasn't obvious before, is that these are all, this element here that's defined by this interval 17679,527 to 17679,727 is inside all of these genes. So the distance here is 0. So of course, all of these are closest. So that means these overlap. So maybe we want to get actually not the closest element including overlapping elements, but the closest element that doesn't overlap. And so now we can use there's another option to closest, this -IO option excludes overlapping elements. And if we do that, now we see that none of these have duplicates, right? Each element, each enhancer in our human enhancers element is now only listed once, except for this one, which actually has a tie where these two are exactly equal in distance. Each one has, we've now eliminated all of these overlapping elements here. So in this case, maybe you want to find only, you don't want to look at enhancers that might be in introns. You want to find only intergenic enhancers, or the closest gene that's upstream or downstream of the enhancer instead of the gene that it's in the intron of. And this now would give you that option. And so you can see as we add these options, the way that our data, the calculations that Bedtools does for even something as simple as determining closest changes, and we get different answers each time. And so this is one reason to use Bedtools, is that although you have to figure out what options you want to specify, it does all these calculations for you and allows you to do all this filtering. And so just as an example, we can look at the help option just using -H for Bedtools closest. And we can see that there's many other options here, right? So for example, if we wanted to do the closest upstream element, or say the closest downstream, so say we wanted to find for each putative enhancer, the gene that was closest downstream of the enhancer, so the enhancer is upstream of the gene. We could use this ID option, which ignores features that are down, we'd use the IU option, so we'd ignore features that are upstream of A, and that would just give us things that are downstream, right? And you can see that in general, there's a pattern here, right? So the -I is ignore something, and here we're going to ignore overlap, here we're going to ignore upstream, and here we're going to ignore downstream. So if we ignore downstream, we get only upstream elements. If we ignore upstream, we get only downstream elements. You notice that there was also some ties that we saw. And so there's an option here for how to handle ties. The default is to report all. And so that is what we saw in the behavior of the examples we looked at, is that every time we had a tie, for example, when we had many overlapping elements, or when we had two elements that were exactly equidistant from a particular enhancer, we got all of them. But you might just, for example, want one thing. So you could choose to report the first tie or the last tie that occurred. And you can scroll through and you can see that there's many other options here, right? You can require strandedness. Of course, this only makes sense if both of your bed files have a strand. So if we scroll back up here, we can see that the gene file, which is defined by these 6 columns here, has a strand information and has names, whereas the enhancer file only has chromosome start and end. So it wouldn't make sense to require strand things on the same or different strand here, because only one of our two bed files has a strand. Okay, so closest is a relatively straightforward operation, although you can see how it gets complicated. Another thing we might want to do is find overlaps between two sets of intervals. So say we have these enhancers that I said were identified in neural crest cells, and we might want to compare them to enhancers identified in some other cell type. So here, for example, we have another bed file named H1 HESC H3K27A chromosome 22.bed, which has H3K27 peaks from embryonic stem cells. So this is another set of putative enhancers, but from a different stem cell. And we might want to find overlaps between these two files. So there's of course, a Bedtools option for this called Bedtools intersect. And then again, it uses an A and a B file. But there's a lot of variation in how this is reported. And this little diagram here that I've taken from the Bedtools website was just showing a few simple options. So let's say we have these A elements up here and these B elements down here. We just want to report intersects. That actually truncates the A element, right? So this blue thing, to only report the things in green that overlap. So the element itself is not reported, right? Not the entire element in A that overlaps, but just the regions of that element that overlap. Sometimes that's what we want, but sometimes we want to know the entire element in A. We just want to know if it overlaps something in B. So there's another option, WA, that reports the entire element, but only reports that element if it overlaps something in B. So in this case, this element here, which doesn't overlap anything in B, right? There's no overlaps here, is not reported on this row. There's another option, -V, which just inverts that. This is like grep-V, if you're familiar with grep. It only reports elements that don't have an overlap. And again, it reports the whole element. So this element here in A is not reported in these first two rows because it doesn't intersect with B. Whereas this element is the only element that's reported in this last row, because with the -V option, we're requiring a lack of intersection instead of the presence of an intersection. Okay, so we can look at some real data here to see how this ends up looking. So here's our here's our initial run, right? With just this is just this first row up here, A intersect B. It reports just chunks of these, of this file, this human enhancers file, that overlap the second file, but it doesn't tell us what they overlap. And notice that it also sometimes reports elements twice, which implies that they overlapped two things, right? So there was probably two elements in here that overlapped this, but these are the same, so they must have been, there must have been, say, two elements here that this element in A was completely contained within. But there's not very much information here. We don't know what it overlaps, right? We don't, you know, we don't know which pieces overlap. So I'm going to use this to look at more information. I'm going to use this WAO option, which is like the WA option, but it only reports a single overlap per element in A. So if this element, sorry, it reports the original element in A, and what it overlaps in B, right? So this is not just the WA, which reports just the overlap, but it also reports what it overlaps in B. And we see here that we get the elements in A here, and then after that, we get these columns for what they overlap. And so now we can see why this thing showed up twice. It overlaps two different elements in B, which themselves overlap each other, right? So there's one element here and one element here that both overlap, and they both overlap this. We also get a list of the length of the overlap. In this case, all of these overlaps, the elements in our B file tend to be much bigger than the elements in our A file. All the elements in our A file are always 200 bases because that's how they were defined. And we can see that this overlap is almost always containing the entire element in A from this last column here, which is the length of the overlap. We can also now see that this B file here is this 5 columns here. So it has a chromosome, a start and end, nothing in the name file, and then it has a score here, but no strand. We also notice now that there's some elements here in A that have no overlap in B. And if we look up here, we can see that these aren't reported, right? We go from 179.15832, which is here, to 1916389, which is here. These ones aren't reported. With the WAO option, we report all elements in A, regardless of whether they have an overlap in B or not. And then we report, for the ones that don't have an overlap, just this stereotypical line with -1s for the numerical values and dots for the text values. And then the length of the overlap is reported as 0. So you can see this can start to get relatively complicated, but now these tools allow you to do a few useful things. So let's say we wanted to now split our human enhancers chromosome 22 file into one set that overlaps with these embryonic stem cell enhancers and one set that doesn't. So you could do that with, with grep or with something like that, right? You could search for these elements that don't overlap and pull them out into a separate file or something like that. But of course, with the -V option, that makes it much simpler, right? We can just use the -V option, report each element in A that does not overlap with an element in B. And this just gives us our list of things that don't overlap. We can see here these two elements, the 18546, 18546, and 18647 show up here, and then we get a bunch more that don't overlap. So we could redirect the output of this instead of to head to a new file that we could call, neural crest specific enhancers. And then if we run this cell, we'll see this is going to show us the, the ones that do overlap, similar to what we showed before. Now, really ideally what we'd want to use here is probably not the WA option, but the option, which actually I'm forgetting off the top of my head. There's another option that reports each element in A only once, no matter what, right? Instead of reporting for each intersection, the element in A, and we'd probably want to use that option for this instead of the WA option. But again, we get a list of the things that do overlap and then the things that don't overlap. And so we could use this to separate things into two different files. Okay, so I'll wrap up in the next 5 minutes or so. A couple more things. There are some Bedtools options that don't compare two files, that instead just work on a single file. And these have a different sort of general format. So I'm going to give you an example of, let's say you wanted to calculate intervals that represented promoters. So for example, here we're using the Bedtools flank command, which creates a new interval that flanks an existing interval. And these Bedtools options that work on single files use a -I instead of a -A and a -B. So we're only looking at this one file. And for many of these, if they're doing operations that require knowing where the start and end position of the genome is, we need to give it a genome file. And this is just a file that lists for each scaffold in your genome. So in this case, it's just chromosome 22. It's just a scaffold name or a chromosome name and then a length. That's all, as a tab separated file. But we need that because for this flank operation, what we're going to do is for each gene, we're going to create a new interval that extends 2,000 base pairs away from the start of the gene. Now, if you had a gene that started on base 562 of a chromosome, then you can't make an interval that extends 2,000 base pairs away from the start because it would go beyond this. It would start before the start of the chromosome. And so we need this genome file to correct for these edge effects. So the options here, so flank, what it does is it takes a bed file of intervals and it creates new intervals on either side of them. The -L interval tells you how big you want the left-hand side interval to be, and the -R interval tells you how big you want the right-hand side interval to be. So in this case, we just want the left-hand side. So we're using -L of 2000 -R of 0. So for positive strand genes, this would mean the upstream region of the gene. Now this gets tricky because some genes are on the negative strand and so everything's inverted. Rather than having to work that out ourselves, we can just use the -S option, which tells Bedtools to treat left and right as strand specific. So on negative strand genes, invert left and right. So we can see what this does. You can see this gives us a new bed file where everything is copied over from this genes bed file, except the start and end positions are different now, right? These start and end positions are the flanking 2kb upstream of the gene, but we copy over the gene IDs here, and we also copy over the strand because we specified that this is a strand specific option. So one of the powerful features of Bedtools is that you can combine different tools with the Unix pipe, which allows the output of one tool to be the input. So what we're going to do here is we're going to Bedtools flank option to make this new bed file of promoters. And then we're going to intersect with these human enhancer bed file to get a set of these human enhancers that lie within promoter regions of these genes. So this first part of this command is exactly what I just ran. But now instead of pipe to the screen with head, I'm going to pipe this to the Bedtools intersect command. So we're going to use as the -A file, we're going to use the enhancers, and as the -B file, we're going to specify a dash, which means user input. So in that case, this is the output of this flank command. And so when we run this, you can see we'll get now, for each one of these elements, we only get the ones that fall within these promoters. And this -U option means we don't care what it intersects or how many times it intersects something in B. We just want to know if it has one or more intersections in B. So print everything in A if it has one or more intersections in B. Okay, so that's just a brief introduction to the sort of two of Bedtools commands, ones that operate on a single bed file, which use this -I option, and ones that operate comparing files. There's many, many, many options in Bedtools. You can also, for example, convert BAM files that have reads mapped to the genome to bed files, which then, so there's a BAM to bed option. which then allows you to do things like compute coverage using Bedtools. There's lots of options, and I think my goal here was not to sort of go through everything you can do with Bedtools, but just give you hopefully maybe a little bit of an intuition for some of the general ways that the program works, to help you kind of figure out how to set it up when you want to do your own things, which could likely be something different. I'm just going to end with a few quick hits, so just like examples of command lines that do certain things. So let's say you wanted to make a bed file of intergenic regions, so regions that are not part of exons or introns, and we have a bed file of genes, and this genes bed file, I should say, is not exons, it's just the start to end position of the gene and includes introns. So there's an option complement that takes a single bed file as input and one of these genome files, and it just inverts it, so it gives you everything the reverse of this. So if these are genes, the output of this would be intergenic regions. And so this would be the intergenic regions on chromosome 22. You might want to, you know, for some sort of permutation test, you might want to randomly permute a set of elements, put them in random places on the genome. There's an option in Bedtools called Shuffle that will do that for you. And it has the ability to either exclude certain regions where you don't want them to land, or include certain regions where you do want them to land. So in this case, I'm going to permute these enhancer regions, but require them to fall outside of genes. And again, I mean, this output is not particularly informative because this is just a list of random regions on the genome. But this is the sort of way you'd set up this command line to do this. Okay, so here's an option of things that are a little bit more complicated. There's a Bedtools option called Bedtools Jacquard that lets you compute a Jacquard similarity index between two sets of intervals, which is basically just a way of measuring how much, using set theory, how much two things overlap. So let's say we wanted to use that to compute with this human embryonic stem cell file. You know, how much do they overlap with promoters? Right, so we have our Bedtools Flink option to create a set of promoters. I'm just piping that into Bedtools sort just to make sure it's sorted appropriately for the Jacquard command, which requires sorted input. And then I'm going to use the -A option from the Jacquard command as our enhancer file, and the -B option is standard input, which is coming from this promoter. So we can do that. I mean, this isn't necessarily particularly meaningful, right? This tells you the intersection, the union, the Jacquard index, which is 0.1, and the number of intersections. This Jacquard index ranges from 0 to 1, so this might seem kind of low, but we can compare that to just totally random regions across the genome. So we're going to use Bedtools random, which will create -n, in this case 743, which is, you know, the number of elements I have in this promoter file. Completely random elements of a certain length. So we could now, instead of asking how much does this human embryonic stem cell enhancer data set overlap with promoters, we could ask how much does it overlap with just random elements in the genome. And you can see here that this Jacquard index is much lower. So of course, this isn't really a formal statistical test, right? There's actually ways using Bedtools that you can compare whether two sets of elements overlap more often than you'd expect by chance. But this just gives you a sense for how you can, you know, potentially string these kinds of things together to start to get at, you know, these kind of pretty complex questions. Okay, so that's all I've got. I hope that was a bit of a whirlwind, but also a useful kind of introduction to the basic framework and sort of conceptual idea is how this Bedtools program is set up, and gives you some ideas for how you can kind of look through the options and figure out how to use this with your own data. And with that, I'm going to stop the recording.